I'm going to talk about some of my PhD work on microclimate variability and butterfly thermal tolerance in the Andes. Ectotherms in the tropics, such as insects, are thought to have narrow thermal limits, potentially increasing their vulnerability to climate change. Because in the tropics, the climate is stable throughout the year, but it changes more rapidly across elevations, creating sharp transition zones. And with warming climates, many taxa have been recorded to somehow expand their elevational range upwards, and habitat degradation in the lowlands, like it's happening in Borneo, is also accelerating this phenomenon. So it's becoming increasingly important to understand the potential of organisms to adapt to altitude, as well as to identify the traits that allow the colonization of these elevations. And a good approach to do this is to study taxa that inhabit large ranges, like Heliconius butterflies do. Some species are very widespread, whereas others are restricted to either the lowlands or the highlands. And in mountains, the physical environment changes a lot over a short space, which increases selection pressures. So given these differences, how can some species, like Heliconius aerato, happily inhabit this whole climate? Today, I'll present results from my PhD, where we tried to understand the climates experienced in the wild and identified phenotypes associated with thriving at high altitude, as well as discuss their vulnerability to climate change and potential future work. Rainforests are incredibly complex habitats, and these butterflies tend to cruise in and out of it, searching for flowers, host plants, or mates. So despite climate change being relatively constant throughout the year in the tropics, climatic variation within forests can be very large. And publicly available data sets are not ideal for understanding this variation, as they tend to be interpolated from weather stations like this one in Ecuador, which are specially designed to minimize the impacts of habitats on readings. So what we did was record temperature and humidity in 28 sites across both sides of the Ecuadorian Andes at high and low elevations. And we did this for a full year with 64 data loggers. Half were placed in the understory and the other half were placed in the subcanopy around 11 meters in height. And with this paired approach, we can really compare the temperature and humidity variation throughout the day in different parts of the forest that the butterflies actually use. So here I'm going to show only a couple of results. First, how mean temperatures change throughout the day. And indeed, we did find that the lowlands here in pink are about 5 degrees hotter than the highlands, but the forest buffers temperatures a lot so that the understory is much cooler than the canopy, especially in the hotter parts of the day. Another physiologically relevant result is the range of temperatures experienced throughout the day. And we found that temperature range is higher in the lowlands, and that the difference between the canopy and the understory is also stronger in the lowlands. However, if we had not gone through this effort of measuring microclimates in the wild, we might have got quite a different picture. So the stars here represent interpolations from WorldClim 2. And as you can see, not only they are higher, which you might expect, but the trend is also the opposite, predicting the temperature range experienced throughout the day is higher in the highlands. So this highlights that to study small organisms, especially in the tropics and especially at high altitude where there's a lot less data to interpolate from, we really need fine-scale microclimate data. So how can these butterflies cope with this range of temperatures in the wild? I tested heat tolerance in the wild of 10 species that had a mix of elevational ranges. And for that, I measured the time it took for butterflies to get knocked out inside a chamber at 40 degrees. So in the y-axis is knockout time, the higher, the more heat tolerant the butterfly is, and the x-axis is different species or populations. And clearly butterflies collected in the lowlands are more tolerant to heat, as you would expect, even within the same species. But the difference is more striking when you compare the altitude specialist versus the low altitude specialist species that are in gray. What was quite shocking was that nearly half of the individuals of the 3 high altitude specialist species collapsed even before the chambers reached 39 degrees, hence all the zeros at the bottom, with temperatures as low as 35. And in our microclimate data, we had found that at least once a week, temperatures go over 30 degrees in the highlands. So these species must be behaviorally adapting to avoid these dangerously hot times of the day. Of course, with climate change increasing temperature extremes, this could be very concerning for these high altitude species. Another very important trait for thermoregulation in many insects is flight, as it's energetically very costly and under storm selection to be optimized. So I wanted to look at wing shape, and we photographed collections from around the range and extracted wing area and aspect ratio. Aspect ratio gives you an idea of how elongated a wing is. So the higher it is, the longer the wings are. And what we found while controlling for phylogeny and other traits is that species inhabiting higher altitudes tend to have rounder wings, so lower aspect ratios. As I mentioned before, some species have wide ranges. So we're curious to know if wing shape also changes within species. And it turns out that they generally do. So in those species with wide ranges, high altitude populations tend to have rounder wings. And we think that it could help deal with the reduced air pressure by increasing lift during flight, but we haven't tested it. So the fact that we find for both traits, heat tolerance and wing shape, varying with altitude within species indicates that they're potentially important at allowing them to cope with climates at high altitude. But to understand whether these traits were genetically fixed in a generation or plastic, we did some common garden rearing in Erato. So we collected fertilized female mothers from high and low altitude, brought them to a common environment, and then reared the caterpillars in the same conditions and in parallel to then test in the offspring, thermal tolerance, and any other relevant trait. So for wild Erato, we have already seen that the lowland individuals here in pink have higher thermal tolerance. But if you bring the mothers to a common environment, their offspring are actually equally thermally tolerant, regardless of what altitude the mothers came from. So this indicates that this trait in the species is quite plastic. What about wing shape? So here, the high altitude populations tend to have rounder wings at high altitude. But when we bring the mothers, the difference are maintained and even get stronger, suggesting that wing shape is at least partially genetic. And a larger study later revealed that wing shape is highly heritable in 2 species, making it a very interesting trait for adaptation to altitude that deserves more study. So let's put these results into context. It's clear to me that the habitat complexity in the tropics leads to large variation in microclimates within forests, but also large buffering potential across elevations. And that both of these patterns are very relevant for small organisms that live in such forests. So we should always include microclimates in vulnerability models. We have found that a widespread species has quite plastic heat tolerances, but the species with narrow ranges, at least in the wild, showed stronger differences in heat tolerance, making them potentially at more risk from climate change. Finally, the ability to adapt genetically with traits such as those involved in flight might decrease vulnerability of species to climate. And a good approach to study that potential of tropical insects to adapt would be to look at cases of contemporary evolution through ongoing invasions, range expansions, or biocontrol introductions. And with that, I'd like to thank all my the collaborators and people that have helped make this project possible, and you for listening. Thank you.